Hey there, future scientists. Welcome back to another exciting microbiology lesson. Today, we're diving into the fascinating world of bacteria, tiny but powerful microorganisms found everywhere, from soil and water to inside our own bodies. We'll explore their morphology, how they look and arrange themselves, and then classify them based on unique features like staining, oxygen needs, and movement. So, let's zoom in and discover this microscopic universe. Bacterial morphology. Before we classify bacteria, let's first understand their shapes and arrangements. 1. Cocci, the round bacteria. Cocci are spherical or oval-shaped bacteria. But they don't just float around randomly, they arrange themselves in different ways. A diplococci, found in pairs, like best friends sticking together. Example: Streptococcus pneumoniae. B. Streptococci, form long chains, like a pearl necklace. Example: Streptococcus pyogenes. C. Staphylococci, clusters like grapes. Example: Staphylococcus aureus. D. Tetrads, arranged in groups of 4. E. Sarcina, form cube-like structures of 8 or more cells, like tiny dice. 2. Bacilli, the rod-shaped bacteria. These bacteria are shaped like tiny rods or cylinders. A single bacillus, just one lonely rod. B. Diplobacilli, rod-shaped bacteria in pairs. C. Streptobacilli, form chains, just like streptococci but in rod form. D. Coccobacilli, a mix between cocci and bacilli, short and chubby rods. 3. Spiral bacteria, the twisted ones. These bacteria have unique spiral shapes. A vibrio, comma-shaped, like a tiny curved bean. Example: Vibrio cholerae. B. Spirillum, rigid spirals with external flagella that help them move. C. Spirochete, flexible spirals with internal flagella, giving them a wiggly, corkscrew motion. Example: Treponema pallidum. Classification of bacteria, on the basis of staining. Now that we've covered shapes, let's classify bacteria based on other important features. Gram staining, the coloring test. Scientists use gram staining to separate bacteria into 2 groups based on their cell walls. A. Gram-positive bacteria have thick peptidoglycan walls that hold onto the purple stain. Example: Staphylococcus aureus. B. Gram-negative bacteria have thinner walls but an extra outer membrane, turning pink after staining. Example: Escherichia coli, E. coli. Oxygen requirements. How do they breathe? Bacteria also differ in how they use oxygen. A. Obligate aerobes need oxygen to survive, just like humans. B. Obligate anaerobes die if exposed to oxygen, imagine being allergic to air. C. Facultative anaerobes can switch between oxygen and no oxygen, super adaptable. D. Microaerophiles like a little oxygen but not too much. E. Aerotolerant anaerobes don't use oxygen, but they don't mind if it's around. Nutritional requirements. What do they eat? Bacteria have different ways of getting food. A. Autotrophs make their own food using carbon dioxide, like plants. B. Heterotrophs depend on organic materials. Basically, they eat what's available. Spore formation, the survival trick. Some bacteria form endospores, tiny protective shells that help them survive extreme conditions like heat, radiation, or harsh chemicals. Think of it as going into hibernation. 2 famous spore-forming bacteria. 1. Bacillus anthracis causes anthrax. 2. Clostridium tetani causes tetanus. Motility. Do they move? Some bacteria have flagella, tail-like structures that help them swim. Their flagella can be arranged in different ways. A. Monotrichous, one tail, like a tiny sperm cell. B. Liphotrichous, a tuft of tails at one end, like a ponytail. C. Amphitrichous, one tail at each end, like a propeller. D. Paratrichous, covered in flagella all around, super speedy. World of bacterial morphology and classification. From their different shapes to how they breathe, eat, move, and survive, bacteria are some of the most diverse and adaptable organisms on the planet. Did you learn something new today? If so, be sure to like, subscribe, and hit the bell icon for more exciting microbiology lessons. And drop a comment below. What's your favorite bacterial shape? Until next time, stay curious, keep learning, and keep exploring the microscopic world.